It looks good.
Okay. Good morning and welcome everybody to day 3 of this Metabolomics workshop. Today we will talk about mass spec imaging and metabolomics with spatial resolution to give a bit of context of where mass spec imaging is lying within all the tools that we have available for metabolism research. We have seen over these 3 days of workshop both global approaches for comprehensive profiling, then how to focus on targeted pathways and validating those changing through targeted metabolomics. But these two methods are mostly in bulk, meaning we sample a certain amount of volume or mass of our sample, and then we measure the average of metabolites in that sample. But what if we want to understand the heterogeneity of metabolism, particularly over space or across a population of cells? Well, in that case, there are other 3 approaches. One being the historically oldest one, mass spec imaging, that has over time evolved in what is called the mass spec imaging at the single cell level resolution. That has been pushed by some lab to the subcellular level with approaches that are able to dissect metabolism at the micrometer range, meaning that it is possible with some very specialized platform to look into the localization of metabolic pathways and where they are occurring over space in the cell. The other approach with a very high resolution in terms of cell, but not in terms of space, is online single-cell metabolomics, where there is a microfluidics apparatus prior to the mass spec and the LC system that separates each cell, and then the extraction hub occurring online enables to measure each cell injected at a time in the mass spec. And in that case, the data looks like more population of cells. More for those familiar with single-cell RNA sequencing, the plots in this case are similar to that allows to identify subpopulations of cells with different metabolic programs active and so on. And then going back to the high throughput approaches without the chromatography that are usually very suitable for big clinical studies where we have thousands of samples. We don't have the ability to retain to invest in time for the chromatography. This all together complemented by stable isotope tracing for confirming and getting further insight on the flux, the directionality of the metabolic changes, and then systems level integration with proteomics, mRNA sequencing, and other omics layers. So today we will focus on mass spec imaging, and we will see an application of that. So what is mass spectrometry imaging? Mass spectrometry imaging aims at detecting metabolites over space. So you can imagine, as reported in this slide, we have a surface that is usually a silicon chip, where we adjust a slice of tissue or the samples of interest, right? This slice of tissue is usually in the order of 5 micrometers or less. And what happens is that in the most common setup, there are different setups, but in the most common setup, we use a mass spectrometer with an ionization source, which is called multi-ionization. What MALDI is a matrix-assisted laser desorption ionization. And what is the difference with what we saw during the other days? So during the other days, we mostly focused on electrospray ionization, right? Where there is a source, there is a sample or a fraction of the sample that is injected over time, a difference in the potential that is causing either an ionization in positive mode or in negative mode. In this case, the setup in the source is different. We have this surface where we have the sample, and then we have a laser, and this incident laser hits the sample, and by hitting the sample with a certain energy, that event is causing the desorption and ionization of the molecules. So we don't have a difference of potential here, but we do have a laser that over space literally goes point by point on the sample and causes the ionization of the molecules. Now, Of course, this is less soft of a technique than electrospionization. It comes with the advantage of having spatial resolution and being very direct on the sample. But in order to cause the diagonalization, the sample per se, the surface of the sample needs to be covered with what is called a matrix. And that's why it's called matrix-assisted laser ionization. What is this matrix? Usually, like there are historically some that are reported in this table, like the DHP, the dihydrobenzoic acid, or HPA, hydroxy picolinic acid. So these are compounds. These are compounds that are distributed on the surface and are able to enable the desorption and ionization. And they do so by either transferring a proton or the electron. So there is an exchange that enables to ionize the metabolites or the molecules in the sample. So these matrices are not only used for small molecules, but they are also used for proteins. Indeed, MALDI actually originated as a technique to measure proteins and peptides. But over time, because operationally in the lab is a very fast approach for measuring it, it has been used also for instance, oligonucleotides, lipids, and for some small molecules. For some small molecules and few metabolites, and the reason for that is because there is a limitation in the application of MALDI as it is for the measurement of metabolites. And that limitation is in the fact that the matrix per se, when it's hit by the laser, ionizes. And where the matrix ionizes is in the low molecular range, in lower than 800 Daltons. What that means is that the noise from the matrix is actually falling in the region where you expect to measure the metabolites, because the metabolites are usually between 50 and 1,200, 100, 500. I would already say from 700 units on, it's usually more lipids and bigger metabolites. So really this region is critical to be able to perform metabolomics. So that's a problem, because we want to have spatial resolution, meaning high resolution over space, but also high sensitivity, because the amount that we will be able to ionize from that spot is very limited, right? So we need high sensitivity, but we are in conditions of having high noise. How do we deal with that? So this type of approach is generally not suitable for comprehensive metabolomics in space, and it needs adjustments that we worked on a time ago in order to make this approach into the ultra-sensitive range. And the way we did that was by developing ionization matrix that was a bit of unique of a kind. And we did it by using gold nanoparticles and functionalizing them with perfluorinated chains, as you can see here, the gold nanoparticles and the perfluorinated chains. So when the laser from the MALDI hits these gold nanoparticles, these functional gold nanoparticles that are distributed over the sample, the energy is first transferred to the gold nanoparticle, then goes to the surface. It's a way softer energy transfer approach as opposed to directly hitting the matrix and transferring to the analytes. So the energy goes from the laser to the gold nanoparticle, transfers to the surface, and releases those chains of perfluorinated chains. By releasing the perfluorinated chains, that act enhances the desorption and ionization of the metabolites. The positive is that with this approach, we can measure in the small molecule range with way less noise than using a classic MALDI matrix, and we can go to ultra-sensitive levels. As you can see here, it's not only that, because it allows us to measure things that normally with a classic MALDI matrix are very difficult to measure, like glutamate, lactate, polar metabolites, together with, for instance, lipids within the same run. So to understand whether such type of approach was actually amenable for biological studies, we made this investigation. It was in the frame of a bigger investigation, but we thought this was interesting enough to have an understanding of both like the spatial resolution of metabolism together with an understanding of whether the technique could indeed could confirm the expected changes. So we took mice under 3 different diets for 1 week. A standard diet, a fiber-depleted diet, and a polysaccharide and fiber-depleted diet. We wanted to chart the effect of diet on the gut metabolism as a non-invasive dietary intervention in health and disease. So this was the starting point to understand whether a diet could indeed change the metabolic distribution of the content in the gut microbiome. We slide the colon at 5 micrometer slices, and then we adjusted these slices on the silicon chip that you saw in the former slide. And so here the objective was to validate whether this NIMS approach, nanostructure initiation or mass spectrometry approach, which is the way to define broadly the category that included perfluorinated gold nanoparticles as a matrix, whether this approach was suitable for charting metabolism over space. So in order to do so, we had to first verify that this approach could satisfy a list of requirements, and particularly it should have not been destructive for the tissue and for the metabolites, right? It should be soft enough for measuring the tissue without creating complete depletion. It should be limited metabolite dislocation, meaning that when we use a hydrophilic matrix, what happens is that because it's dissolved on the surface of the sample, it can, if it's not evaporated fast, it can dislocate the metabolites, right? So because we want the high resolution, we also wanted something that would not perturb that localization over space. An easy protocol to access it. So to synthesize this matrix, low matrix interference, heterogeneous coverage, because we would, in this type of studies, we would not be able to go back and perform multiple measurements, and enable comparative studies. So for the first requirement, we did the measurements either in presence of a classic multi-matrix and a fluorinated matrix. The perfluorinated matrix here, this is the intact tissue only hit by the laser, and these are the gold nanoparticles after being hit by the laser, right? So as you can see here, the laser per se does not cause ablation of the tissue, but when we add the classic multi-matrix, we have complete depletion of it. When we use the fluorinated gold nanoparticles, this detection is way softer. You can see it from this image. And we also observed that the laser per se is causing expansion of those nanoparticles, meaning almost, I would not say complete melting, but clearly the energy is transferred to the nanoparticle. Limited metabolite dislocation. So these nanoparticles are perfluorinated, right? Some of you might be familiar with perfluorinated compounds. Do you know where we all have them in our houses, actually? Do you, have you ever heard of them, perfluorinated? Okay, so have you ever heard of Teflon? Okay, the Teflon is made of perfluorinated compounds. And what makes it unique is that it's not hydrophilic, it's not hydrophobic. And that is the reason why it doesn't stick. Yes, yeah. And so the surface of these gold nanoparticles, because it's perfluorinated, you can imagine it that it has exactly those characteristics, has the capacity of being distributed on the sample. It evaporates very fast because the solvent is perfluorohexane. So perfluorohexane is a solvent that evaporates immediately, almost immediately. So they are distributed on the sample. The solvent evaporates very fast, but at the same time, because they are not hydrophilic and not lipophilic, they don't dissolve. They don't mix with the sample. They just adjust on it. And there is an easy protocol for synthesizing them. This is how the gold nanoparticle looks like when when we buy it, and then this is the simple reaction to synthesize it in the lab. It's a 3-day process. It's quite simple, so it does not need like a full organic chemistry lab. Okay, so low matrix interference. So this is what we will see in the next slides through looking into the data from this study, and the heterogeneous coverage. So for this, we took these mice, 3 conditions. We had these colon slices, we covered with fluorinated gold nanoparticles, and then we performed MALDI analysis with the laser in the source to obtain images as the one reported here. So from this study, what we saw is that with a step resolution of about 5 micrometers, for each point of the image where the laser was performing the ionization and the mass spec detection, we measured about 1,000 metabolic features, not metabolites, right? At that point, those metabolic features, we computationally annotated them against the Matlin database, and that annotation provided us with about 80 metabolites, which were distributed in this way across metabolic pathways. And the interesting part here, which is very difficult to achieve with like very classic MALDI, is that we could measure not only lipids, but also bile-acids, but also polar central carbon metabolites, right? Carbohydrates, amino acids, nucleotides. So that was very interesting to us, and particularly because mass spec imaging has the limitation of being usually very biased towards lipid detection. So being able to tap into more polar central carbon metabolism was really an advantage. Now, one limitation of mass spec imaging is that usually very few of these metabolites can be confirmed by fragmentation experiments by mass spec imaging. And the reason is because the sensitivity required is so high that when we switch to fragmentation experiments, only for very few of these is possible to isolate enough precursor to actually get good sensitivity on the fragmentation. So generally what is done in this type of studies, when there is some metabolites that are changing and are interesting, they are charted through mass spec imaging, but their identity is confirmed by doing selective sampling of parts of the sample and then submitting them to classic in bulk measurements. So yes, so but meaning that this approach allows you to find areas in the tissue that are metabolically different, but then when it comes to the identification of the metabolites through mass spec from fragmentation experiments, then that is usually done by selectively sampling from those different areas that you identify through mass spec imaging, right? So going back to the sample, selectively sampling from those areas, and then measuring and doing fragmentation experiments on a classic LC-MS system, right? Because you have better sensitivity, you can get better MS2, and yeah. For few metabolites like glutathione, like very high concentration in the cell, it can be possible to fragment directly with mass spec imaging. Some lipids as well, the very abundant ones, but not all of them. So it's that is a bit of a limitation as opposed to LC-MS. So we started looking at the coverage, and we started plotting the intensities. And as you can see here, so the white part is the lumen of the gut, and the inner part is the content. You can see that within the same analysis from the same mass spectra, we can measure sulfuric acid, glucose, but also hydroxycortisol, glucosamine, lipids. So this was really the unique aspect that enabled us to have like a more comprehensive overview. And then when we looked into comparative studies, meaning are we able through this approach to actually capture changes at the metabolic level with spatial resolution and at the same time having a semi-quantitative measurement of those changes. And the answer was yes. So because when we deplete carbohydrates and polysaccharides, what happens is that they are usually broken down by the gut microbiome in small chain fatty acids. Your expectation would be that for the carbohydrates and polysaccharides depleted diets, you would expect a decrease in that metabolism, and that's exactly what we detected. Any questions so far? No. Okay. And this is a summary slide of how does the tool and resource ecosystem look like for mass spec imaging and single cell metabolomics. So far, this is still work in progress. There is a lot of like, this is, I would say, the frontier of where metabolomics is standing now, both method-wise, technically, and development. So sample preparation, there are different ways to prepare the cells for single cell metabolomics. One is online workflows, meaning they separate the cells and then they have microfluidics that allows that. But also there is an approach which is called MAMS, where there is a chip that has allocations for each cell, and by means of that geometric allocations, it enables to separate, and then with MALDI knowing where each single cell is present and where the MALDI has to hit in order to make measurements for these single cells. Spatial resolution. So in the techniques that I showed you, in the images that I showed you, we were able to go down to 5 micrometer. But there are some limitations there, and those are technical, both for the step of the laser in the source, literally like how it's designed to move for each single spot that is measured, but also the diameter of the laser, of the incident laser, because depending on the energy, that laser will have a certain diameter. And in this type of measurements, the diameter of the laser is comparable to the resolution that you are trying to achieve. So there are approaches that focus that the same amount of energy in a smaller diameter. It's work in progress, but that is also something that I would say makes a multi-instrument, a classic multi-instrument, not always amenable to go down to the single cell level without having structural adjustments to that. Same secondary ion mass spectrometry, where there is a secondary flow of ions, so it works with ion clusters and causes the ionization of the molecules. It SIMS can go down to 300 nanometers, so better resolution. It's available in very few labs in the world, and usually these are labs that are specialized in single cell proteomics and metabolomics. So the best idea, if your lab does not have this type of approaches, because they are so specialized, is if you really need them for your project, is trying to collaborate, right? Or see whether there is a possibility of having those measurements done by, unless you want to focus for many years on, developing a platform that is dedicated to single cell metabolomics. Then, of course, these metabolites, we have seen the pathway enrichment tools. So from, in this case, because we usually work with metabolic futures, with annotated metabolic futures, the most natural, like the first line way to look into this data is by looking into pathways, because we do not have usually the MS2. And I would say the literature here has a bit of a consensus that because it's the frontier and the limitations are known on obtaining those MS2 from this type of measurements, there is so the expectation for reaching the 4th level of identification are usually lower as long as it's clearly disclosed that it's an annotation, that is not a full identification. And the throughput. So the throughput, because as we go down to single cell levels, in order to have data that are not only representative of the heterogeneity of the system that we are studying at the single cell level to make it to make it robust enough to perform these studies, we also need a certain statistical power in our analysis. So the throughput is really important. So this all again is enabled by bioinformatics. There are some databases in this case that are dedicated either to mass spec imaging or to single cell and to single cell data. GNPS and Metlin are always like good resources here for annotating and for the identification if you have MS2. But I would say Metaspace here is the database where you can have a preliminary annotation of your mass spec imaging data, but also they do have data sets from other studies in the world that have spatial resolution. So very often, if you're interested in seeing if there are data available acquired for us, either your system, your experimental conditions, or similar, and you want to reanalyze them, then Metaspace is the database for those needs. And of course, this can be integrated with other omics. At the single cell level, I would say there is more maturity in terms of single cell transcriptomics, right? Like we have seen RNA sequencing at the single cell level, both with spatial resolution or at the level of population of cells. Is a technique that now is quite it's quite common to find in also with collaborators. So that's definitely something that can be integrated having a combined score of heterogeneity in metabolic pathways, but also single cell proteomics is a very vibrant field. There is both transcriptional, there is both spatial resolution proteomics, but also like more single cell targeted proteomics. So that's also another field that has very interesting updates. And of course, combined with flow cytometry experiments that are at the single cell level. And so there are many options. This all enables single cell metabolomic subcellular measurements. There has been a couple of very good publications recently about the subcellular compartmentalization of metabolism and the organization of some metabolic pathways that historically were always thought to be more distributed in the cell, but now by use of subcellular metabolomics with SIMS, they were able, for instance, to demonstrate that purine synthesis is actually arranged in structures that are called purinosomes. So these are, very interesting findings that are at the forefront of biochemistry that are enabled and unlocked from techniques that are definitely a bit less common than classic metabolomics, but they really allow to push biochemistry forward and understand metabolism at a new level of resolution. Systems level interpretations and systems biology, right? So this is always the approach also in this case, just different level of resolution and understanding within cells and across populations of cells. And so this is for you to have an idea of how the protocol for making the fluorinated gold nanoparticles and utilizing them for mass spec imaging looks like, right? So it's more feasible than it sounds from just looking at the images. So there is a 3-day organic chemistry synthesis step. Then after that, that matrix can be used directly on the sample for analysis. This is a mass spec with a multi source that this is the one that we were using because it's amenable for changes in the source that can accommodate the silicon chips. So this is the silicon buffer where we mount the tissue that has been first sectioned in a cryostat. And so because the silicon buffer has so it needs to be primed. This needs to be prepared with a piranha etching solution that allows the tissue to be mounted properly on the silicon chip. And then from there, this chip is moved inside the source of the mass spec. And then, of course, there is a method that will determine what is the resolution of the space, what is the laser step, and what type of mass range will be detected with which resolution and so on, and that will generate this information which spatially resolve the data matrices for analysis. Okay, so this is the end of our workshop. Going back, this is a summary. You can find these slides both in the recording online, but if you want the full slides, just send me an email. I'm happy to share also. We covered some of what we can do with these approaches, but both in terms of data integration, interpretation, and also adjusting these workflows for specific needs in your projects. If you feel that there is some parts of these workflows that can be useful for your projects, don't hesitate to send me an email. We can discuss. I'm happy to help you with a bit more training. Thank you so much.